Excellent. Hello, everyone. Nice to meet you. Thanks, Steve, for the invite here today. My name is Nikolaou. I'm the head of cell biology at Pixlbio, and what I'm going to talk over the next 15 to 20 minutes is our platform focusing on iPSC-derived hepatocytes, why we have it, what are the benefits, and quite frankly, I would like to focus on a project that we started about 3 years ago with Atelerix when we first started as well, which has led to a very successful partnership and the product that we are now offering as a team. But a few words about who we are. Pixlbio is actually a strategic merger between two different companies that came together about 9 months ago. In the UK side, we had Definigen, which was a company focusing on the development of iPSC-derived hepatocytes from human somatic cells. And our team in the UK has been focusing on the generation of liver models, intestinal organoids, and anything that has to do with liver biology and disease modeling overall. And we have been offering ourselves for drug toxicity studies, for drug discovery, and of course, anything that has to do with large-scale screening. So in July, we decided to partner with Fenaros. It is a spin-out company from the University of Uppsala, and they are actually our AI experts. So what they perform is cell painting in large scale, matched with AI, in order to generate large-scale toxicity screening platforms for both disease modeling, for both drug screening, and anything that has to do with mechanism of action studies. And as you see here, we now operate as one team and one platform. Both sides still operate independently to some extent. So as a company, as a UK side, we still offer our wild-type hepatocytes, our disease lines to our clients, so we operate as we used to. At the same time, our Swedish side is cell-type agnostic, which means that they have a library of more than 30 immortalized cell lines where they perform cell painting for drug discovery. But of course, now as a team, we feed, as a UK scientist, the Uppsala platform with our hepatocytes in order to perform, as I said, drug discovery and large-scale toxicity screening. However, for the sake of this presentation today, I will focus on what we did with Steve and Atelerix team 3 years ago. And for this reason, I would like to focus exclusively on our hepatocytes, on our product. But first, I would like to say describe why we are developing iPSC-derived hepatocytes. So in the liver space, there are 3 different models that people are using in order to perform metabolic studies. Normally, we have hepatocellular carcinoma cell lines. So these are liver cancer cell lines that scientists are using in order to predict toxicity, drug discovery, and anything that has to do with human metabolism. However, these cells come with significant disadvantages when it comes to cell health because they are cancerous. And I think this, the title speaks volumes in this regard. To overcome those limitations that those hepatocellular carcinoma cell lines come with, people are using primary human hepatocytes, which is now considered the gold standard model when it comes to liver metabolism. However, these cells also come with significant disadvantages, and these mainly focus on the fact that they stay active for just a few days post-culture. You come across significant variation across different donors, and quite frankly, these are quite expensive because you understand that these have been isolated from human livers, so availability is limited. And here we come as a company with our iPSC-derived hepatocytes, because what we do, we combine the expansion capacity and the stability of the liver cancer cell lines alongside the functionality that one would expect from primary human hepatocytes. And in particular, over the last few years, we have optimized our iPSC-derived hepatocyte platform, and now our cells demonstrate way higher functionality compared to cancer cell lines, comparable to primary human hepatocytes. And on top of all the functionality benefits that we come with, we also offer unique benefits in large-scale screening, including the capture of donor-to-donor diversity. That means that we can take any somatic cell from any human being on the planet, we can reprogram them into iPSCs, we can bring them back into the pluripotent status, and then we can differentiate into hepatocytes. And by doing that, we can actually track population diversity in a dish. Our cells, unlike primary human hepatocytes, are extremely stable, and I'm going to show you some data later. And when I'm saying stable, we can keep them in culture as a 2D monolayer, as a 2D platform for up to 35 days. So that is particularly important when it comes to drugs with either slow clearance or with drugs that are difficult to metabolize. And third, a unique benefit when it comes to iPSC-derived systems is the ability to generate disease models. And I'm talking about the ability to introduce mutations or SNPs that people see in the general population that you can't find with primary human hepatocytes. So what we do, we introduce this through gene editing, and then we expand the cells, and we can use that for drug discovery studies, as I said earlier. And the last point, as you see here, is now we're offering this to the outside world. There are a lot of companies out there that have been focusing on the development of iPSC-derived hepatocytes. And most of them have been focusing on the characterization of those cells by just checking the hepatocyte markers. Liver is the central metabolic organ in the human body. It performs more than 500 metabolic actions. But most of them can be scaled down into 5 different areas. And these will be the hepatocyte transporters. It can be secretion of albumin and proteins. It is focusing on urea secretion. So urea is actually when proteins are catabolized towards non-toxic metabolites, and they are excreted through urine. And most importantly, and I will talk about that in the next slide, liver is the central organ when it comes to the metabolism of drugs. So when we have any drug or any xenobiotic that we take exogenously, everything goes to the liver, and through different metabolic pathways, those are metabolized, they are inactivated, and then they are secreted in the blood flow and get out of the body through urine. So for us, it was quite important to demonstrate that our cells have all these metabolic pathways active. We wanted to show that our cells were better to cancer cell lines and that they were comparable to primary human hepatocytes. This is a quite busy slide, I understand that, but what I would like you to remember and take home from this slide is that our cells express all the liver maturity markers comparable to what we see in primary human hepatocytes. Albumin, alpha-1 antitrypsin, HNF4 alpha, these are markers that someone should expect from a primary human hepatocyte. All of them are comparable with our cells. Our cells secrete albumin to really high levels. Again, we wanted to compare to the gold standard. We wanted to compare to primary human hepatocytes. That's why all of our data are based on primary human hepatocytes. So you see that our cells secrete albumin over time. This stays stable for at least 24 days upon culture. Our cells have a fully functional urea cycle, which means that we can identify urine. Urine a urea in particular is actually a metabolite that is used in drug discovery and toxicity studies. So differences in urea is highly related to how a drug is metabolized within the liver. And this slide in particular is focusing on that drug toxicity related metabolic pathways that we were interested in. Our cells had really high levels of phase 1 metabolism, a variety of different enzymes that their expression was comparable to primary human hepatocytes, as you see here. As a company, and this goes for the biologists mainly, mRNA levels are beautiful and they give evidence of how a cell should work. But as a liver biologist, we were always focusing on functionality. And functionality can be tracked either through protein expression or through actual metabolic analysis. So for us, it was quite important to identify that all these enzymes that should be in a primary human hepatocyte and in our cells are behaving as they should do. So here you see a panel of different enzyme-related studies where we treated ourselves with different drugs. So these are drugs that people are using on their daily life, and those drugs should be metabolized by those enzymes that are in the liver. And here you see that across increasing concentrations of those drug treatments, we were able to detect the CYP1, the CYP-related metabolites. So in the case of midazolam is metabolized by CYP3A4, and what we could detect in the media of our cells was the product of CYP3A4, 1-hydroxymidazolam. And the same was applied to the rest of the data. And last but not least, for us, it was always quite important to identify that the cells have also phase 2 metabolism. Going back to liver biology, phase 1 metabolism is focusing on the detoxification of the drugs. So drugs are toxic. They enter the cell, phase 1 metabolic pathways inactivate them. However, those products need to be taken out of the cell and excreted to the blood flow. And this is what phase 2 metabolism does. So for us, it was crucial to identify that all the different enzymes that are responsible for those pathways are there. And this is what this graph shows here. We have like really nice expression data of genes involved in the UGT pathways, in the GST pathways, in the sulfotransferase pathways. And similar to what we have done here, we were also able to detect those phase 2 metabolites by mass spectrometry. And quite frankly, this is a study that we performed in collaboration with AstraZeneca in Sweden. And before moving to the data that we generate in partnership with Atelerix, these are some of the additional benefits that you can track with iPSC-derived hepatocytes. As I said, you can investigate donor diversity. At Pixlbio, we have iPSCs from three different human donors, and all of them can differentiate really nicely towards hepatocytes, but all of them demonstrate differences when it comes to their phase 1 metabolic profile. And those differences here with regards to the different SIPs is exactly what we are interested in. We are interested in this donor-to-donor diversity that we see with regards to the expression profiles, because some drugs are metabolized better compared to others based on the humans. Prolonged cell culture stability. As I said, our cells can remain stable for at least 34 days, which is quite important, as I said, for all those drugs that we are interested in as a company. And disease modeling. As a company, we have developed a series of both monogenic liver disease models as well as multifactorial disease models. Fatty liver disease is one of the most popular diseases in the liver field. It is a metabolic disease, and with our cells, we can demonstrate different aspects of the metabolic disease pathway, including lipid accumulation. So when we load our cells with lipids, they accumulate fat. We see instances of insulin resistance, which is something that we observe in patients with metabolic syndrome. And of course, we have increased inflammation, so our cells can get inflamed. And these are the metabolic, the monogenic disease models that I said that we have developed as a company. And unlike other studies, our cryopreserved product has worked quite well. So we are offering this as a cryopreserved product. We also offer this to our clients as in-house screening services, and it has worked amazingly. However, there is one significant limitation when it comes to the cryopreserved product for us, and that is not the functionality, but the duration of the treatment. As soon as the cells are thawed, they need to stay in culture for at least 2 weeks before our clients perform any studies on those cells. And quite frankly, we have had clients who are saying, this is too long. We want to take the cells out of the freezer, we want to thaw them, and after a few days, we want to do the experiments. 2 weeks is too long for us. And this is where Atelerix came to the picture. Because what we did was to evaluate in-house the Atelerix technology. And again, this is something that started, Steve, about 3 years ago. It's been quite a while now. So it's quite developed, which is what we're really proud of. So this is the encapsulation method that Atelerix developed back in the day. 3 different gelation steps in order to protect the cells. And once the gelation has been completed, by using different reagents, we dissociate the gels, and then we bring the cells back to how they are in order to perform the experiments we want to perform. And we performed a case study in-house trying to mimic how that would be if a client was receiving live plates from the UK across the world. So this is a very simplified schematic of the multi-well plate that we used. We had like 60 wells in a 96-well plate. Half of the plate was with non-stored cells with just media, and the other half of the plate was cells with the gelation system that Atelerix developed. We tried to mimic the time points. So for us, most of our clients are based in the US. So, and we know that it takes 3 to 4 days for the cells to arrive from the UK to the States. So we're saying, okay, let's see how the cells will behave after 72 hours. Let's see how the cells behave after 96 hours. And of course, we had another plate in parallel where it was not touched at all. That would be our control. And in-house, what we performed was a variety of different assays in order to evaluate the system. So we checked cell viability using Press2Blue. We checked hepatocyte functionality morphologically by mRNA expression, qPCR, and of course, the most important bit for us, protein expression. And here comes the data. So we kept our cells for 72 hours and for 96 hours, pre and post encapsulation. And as you can see, morphologically, we had no differences. So the Atelerix system was not detrimentally affecting cell morphology. The cells were still attached. They were demonstrating the cobblestone-like morphology that we wanted. But most importantly for us, cell viability wasn't affected, which means that the cells were still active, metabolically active. So you see that after 72 and 96 hours, there were no significant differences between our non-stored cells, the non-encapsulated, and the encapsulated cells. And what about hepatocyte functionality? We started with the mRNA levels of hepatocyte markers, albumin, alpha-1 antitrypsin, hepatocyte nuclear factor 4 alpha. We had our control, the untouched plate, 72 hours, 96 hours between non-encapsulated and encapsulated cells. And as you see, we observed no significant differences between encapsulation and non-storage, which was great because that was exactly what we wanted to avoid, since hepatocyte is mainly affected at mRNA level. So any differences will be first shown at mRNA level. And the problem with hepatocytes is that they are extremely sensitive to any differences and any changes in the environment. I would like to highlight here that everything was kept in 37 degrees. We also performed the same design of experiment into room temperature. Quite a fun experiment. We kept in the cells into a polystyrene box for 3 days, so we're waiting to see how the cells would respond. But it doesn't have to do with the system, it has to do with the cells. Hepatocytes simply don't like being outside the incubator. But again, the most important bit for us was protein expression. So matching the mRNA data, we performed immunocytochemistry-related experiments to evaluate the expression levels of our cells in the system. So albumin, hepatocyte nuclear 4 factor alpha, in red and green respectively. And I think this picture speaks for itself. You see no differences in the expression and the percentage of positive cells between the two different systems. And that was extremely promising because it was really this data that we used to go to our clients in the US in particular and say, you know what, we have an alternative system in place that we can use in case you don't like cryopreserved vials. And it was successful because we have completely successfully 3 different studies by using the Atelerix system with different clients across the pond. The first case study was an evaluation of our Pixlbio using the Atelerix system for a company that is located in Boston, Massachusetts, in the US. The second study was again for Boston. The difference between the second and the first is that in this case, we also used diseased cells at the same time. So for us, it was quite important because now we could evaluate that not only healthy cells can be used successfully to move to the US, but also diseased models. And in this case, we use the urea cycle disorder disease cell that we have developed. And more recently, it was like a closer trip, a shorter trip. So we used the Atelerix system to transfer cells from Cambridge to Stevenage. And that was actually for another cell type. It was for our hepatic stellate cells. And for us, that was quite important because that by using the Atelerix system, we avoided a spontaneous activation of stellate cells, which is quite important for us. What I would like to highlight here is that this is a partnership between 3 different companies. It was Definigen, Pixlbio, as we call it now, Atelerix, and we also had Cellbox. As our cells are extremely sensitive to those temperatures, we had to find a solution in order to transfer those plates into the US without compromising temperature or CO2 environment. And this is what Cellbox does. They have developed this box, hence Cellbox. You can put 96 well plates in there. It is CO2, it is temperature controlled. CO2 comes from dry ice, which I find personally quite clever. And you can ship the whole box into the US or at any other point in the world successfully. So the client receives the cells from the box comes back to the UK, and everyone is happy as Larry. So again, we are really proud because that was a triangle partnership between 3 different companies of similar size, so we should be quite proud of. So to finalize, I hope that I have shown you that our cells are superior to liver cancer cell lines. They are comparable to primary human hepatocytes. They offer a variety of benefits, including large-scale screening, donor-donor diversity, long-term drug effects, disease modeling, and most importantly, for the sake of the presentation today, that the encapsulation system using the Atelerix technology does not compromise the quality of our cells, and that can be used in order to transfer cells all over the world for drug toxicity and discovery studies. Thank you very much for your attention, and I'm happy to take any questions.